So I've mentioned TCRs and BCRs and T cells and B cells, but where do these actually come from? So both T cells and B cells originate from hemolytic stem cells, or stem cells if you just want to be a bit short. These then develop into a lymphoid progenitor or progenitor, and then the separate differentiation begins. So a B cell matures partially in the bone marrow. To be clear, I do tend to sort of like shorten bone marrow to BM. So for further reference, if you see BM means bone marrow. Partial maturation of B cells occurs in the bone marrow. This is when it expresses the first antibody, which is IgM. These are membrane-bound receptors that are around on the B cell. Full maturation occurs in the spleen, and this is when another antibody is expressed, and this is the IgD, which is the D B cell. D antibody even. When it's fully mature, it's released and it's allowed to circulate in the blood and the lymph as a mature but naive B cell. It's naive because it's yet to find a complementary antigen. This is what it's circulating for. As soon as it finds one, it binds, gets activated and differentiates into plasma cells. Upon this differentiation, it stops dividing and it starts secreting these soluble antibodies. So they're no longer bound to the membrane, they just sort of like can float about in the serum. These mature cells can then die within 1 to 2 weeks, but some are saved as immunological memory cells, so they provide long-term memory. That means if you get another infection, that it can remake the same antibodies. T cells, these again from stem cells and lymphoid progenitors, but maturation of T cells occurs in the thymus. When they're in the thymus, they are referred to as thymocytes. So T cells which are undergoing maturation in the thymus are called thymocytes. And during maturation, they go through two sort of main things, one of them which is a bit complicated and I'll go into. So the first one is they rearrange the T cell the T cell receptor on the membrane. The next thing is that they go through something called central tolerance. And this is composed of positive and negative selection. So, positive selection occurs in the thymic cortex. This relies on a T cell binding to self cells. So, the T cell has to bind to self MHC molecules on the thymic epithelial cells. I don't know why I've got whatever that word is. Thymic epithelial cells. I'll just go with that. You know what I mean. The epithelial cells of the thymus. If they don't bind, then they die by apoptosis. So positive selection, the T cells have to bind to the MHC, otherwise they die. Then you go on to negative selection. This occurs in the thymic medulla and it's sort of the opposite. If the T cell binds to a self-MHC molecule, then they're selected against and killed by apoptosis. I know, it's a bit annoying and complicated. But how do these cells bind? Well, you can have different types of T cells. So you can have a double negative T cell. This is when they don't have molecules on them. These molecules are called the CD4 or the CD8 molecule. So a double negative cell has neither of these. You can have a single positive T cell, which has either a CD4 or a CD8 molecule on there. Or you can have a double positive T cell, which has a CD4 and a CD8 molecule on there. But what do these CD4 and CD8 molecules do? Well, the CD4 molecule allows it to bind to MHC2 molecule. If it's a CD8 molecule on there, then it allows it to bind to a CD1 molecule. I know it's a bit complicated. So you have your MHC molecules, and there are two different variations of them. If they have the CD4 or the CD8 present, it can bind to them. A good way to remember which binds to which is the rule of 8. So your CD4 binds to your MHC2. That is because 4 times 2 equals 8. Your CD8 binds to a MHC1. That is because 8 times 1 equals 8. The rule of 8. There you go. A little bit of help there for complications. But back to maturation. Again, when it's gone through this central tolerance, you have a naive but mature T cell. These again patrol the lymph looking for complementary antigen presenting cell antigens in secondary lymph organs. When it binds to these, they become activated and can go through numerous processes.